All right, guys. I know we weren't able to meet on Wednesday for the class because of the weather. So I basically put this together. I'm gonna be doing the presentation online, so you guys can listen. My idea with this is to focus more on things you should expect on the exam on Monday, of course. But of course, at the same time, give you the general idea. By now, everyone should have read this chapter. You should have taken the quiz, so you should pretty much have a general idea of it. So we'll try to focus on the exams, and I put in here the slide which states where the multiple choice questions are coming from, an essay. So it should pretty much set you up well for the exam. All right, so once again, there are times where, oh, ignore that doorbell sound. There are times where I'm gonna go through a slide really quick. Don't worry about it. I just want to try to focus in on the stuff I need you to remember. Okay. All right. So senses, of course, if we were in class, I would have asked you the standard question, which is, which of these guys would you like to be? Of course, hopefully not this one. Maybe this one. Pretty cool. Yeah, definitely this one, right? Just kidding. But yeah, senses, you need them. Basically, that's what connects you to the world. And we have the sensory receptors, which basically how you get information from the outside, inside, wherever it is, that's then converted into some form of information for your brain, right? So those information you're getting from the outside is considered as a stimuli, and the action of converting that stimuli into an electric energy, or more like an action potential for your brain to understand, is called transduction. So they're transducers that convert the stimulus energy into electrical energy. So for like the example of your eyes, your optic nerve will be a transducer, right? It takes the eye the information from the retina and converts it into action potential, which goes to the brain for interpretation. Of course, the question I ask with this is usually pretty straightforward, which is a question of definition basically. If I could say dash converts stimulus energy into electrical energy, and I give you options, transduction and different stuff like that. So as long as you understand what a transducer is, you should be good with this. And next slide. The other part to it is you definitely have the sensory receptors, but the region, the area where you get that information is called the receptive field, right? So it's more like the endings, the distribution area of your endings. You could have a large receptive field or a small receptive field, but that distribution area is basically what I need you to remember, the terminology receptive field. And question here usually is basically a definition based question, right? So it should be pretty straightforward as well. It's funny, if I was in class, this would have been the part where I ask you guys, can you hear me at the back? And of course, there's no one at the back now, alright. So, sensation is generally a stimulus you're consciously aware of, right? So, if you conscious of it, it's considered a sensation, and it has to get into your cerebral cortex for this to happen. The different information that comes in is categorized into four different things, which is the modality, the location, intensity, and duration, right? And I do like to ask this kind of test, more like the receptor, the information provided to the CNS by the receptor is categorized into this, right? And I must tell you, I do try to trick people on this, right? This is one of the parts where I test your spelling out. Instead of modality, I could write modality, something like that. So make sure you pay attention to this. So you have modality, you have the location, you have the intensity, and you have the duration. So for modality, this is basically just saying this is the type of stimulus you get, right? Is it an optic? Is it auditory? And location is saying this is the area, the receptive area that's getting the information is coming from, right? So if you have pain in your leg, that way your brain can tell it's coming from the leg, different parts like that. And the intensity, as you expect, is how intense basically the stimulus is, and this depends on how many nerve signals you're getting, right? Because remember, a nerve, the action potential is all or nothing. So how much of the signals you're getting would determine the intensity, and the duration. And the duration, basically, we're talking about how long it takes, right? And this brings us into adaptation, which is decreased sensitivity. You walk into a room that stinks. Oh my goodness, it stinks. But if you stay there for a while, after a while, it doesn't stink that bad anymore. Doesn't mean the stimulus is not coming in. It just means you have become like adapted to it. Your sensitivity to it has decreased, right? So, pretty straightforward question, typically around here as well, adaptation. And you should know the difference between tonic receptors and phasic receptors. So, ideally, the tonic receptors, like you're maintaining a specific tone, right? So, it doesn't really adapt, well, it's limited adaptation. So, you could be getting a stimulus for days and you're still feeling it. Like, let's say you have a pain in your tooth, for example, so like wisdom tooth or something like that, you're having that pain. Until you address it, it stays there, right? You can you can you can get, well, not really get used to it. You can handle it better, but you're always aware the pain is there. As opposed to a phasic receptor where you adapt rapidly, and or if it continues, you don't notice it that much anymore until there's a new stimuli or increased stimuli to bring about it. So, like a pressure receptor, you've been sitting down, maybe you sit down on something that's hard, and at first you're like, oh, uncomfortable, but if you sit down long enough, after a while, it just feels normal. Excuse me. So, another thing I need you to remember is the classification of sensory receptors by distribution, the stimulus origin, and the stimulus modality. Right? So, distribution wise, we're usually talking about general versus special. Easy to remember. You just need to remember all the special ones, and everything else will be general, right? So, like the special senses you have, we're talking about olfaction, gustation, vision, auditory, and equilibrium, right? So, smell, taste, sight, hearing, and balance. And the other senses are basically considered general, be it somatic or visceral. And the other part, which is the stimulus origin, you're thinking external receptors, which is saying your stimulus is coming from the outside, or internal receptors, which is saying it's from within, like your organs, your internal organs, and proprioceptors, which is basically trying to balance out, which is you trying to balance out your body, your limb movements, trying to see where you are in space, basically. And the third part, which is modality of stimulus, um, which I do ask on the essay question. Usually, I expect you to know the five types of modality of stimulus. I expect you to know what they detect. I expect you to be able to give me a valid example of something that uses that, right? So let's say, for example, you have chemoreceptors. I want you to be able to tell me chemoreceptors, they detect chemicals, and an example would be smell of food. It could be smell of food, yeah, something like that. But something, or taste, your taste buds should be a good example. So we have chemoreceptors, we have thermoreceptors, we have photoreceptors, we have mechanoreceptors and nociceptors, right? So chemical, heat, temperature change, photoreceptors, light, mechanoreceptors, we're talking about pressure, and nociceptors, we're talking about pain, right? Once again, nociceptors would be a good tonic receptor. No, this is usually the part where I say, is there any question? But I'm sure if you have any question, you can email me. All right. So, like a multiple choice question in this regard would be a classification of the receptors. Like the example of the eyes, you can say based on distribution, it's special sense. Based on the location or the origin, it's external, and modality will be photoreceptor, right? So I would give you a question and say something like the nose, which are the sub classifications based on distribution, origin of stimulus, and modality. All right, we'll just talk about a few receptors here when you look at the tactile receptors. So, of course, tactile receptors are mechanoreceptors, which is basically you feel that change, like or pressure or towards the cell membrane, right? So, distortion of cell membrane. When you look at the tactile receptors, you can see here in the skin, we have different types. So you have the unencapsulated and you have the encapsulated. So unencapsulated, as you can see here, we basically have like free nerve endings. And an example of the capsulated, you can see there's a capsule around it right there, right there. So you can see a couple of unencapsulated, encapsulated. So I'm gonna go through this a little bit fast here. I just need you to see the different types. So unencapsulated, you have the free nerve endings, which is basically the simplest tactile receptor. It's just like you expect a nerve ending picking up stuff. And then the other type you have is the root hair plexus, which basically it's a plexus wraps around the root hair and it helps detect hair displacement. So if there's a movement in your hair and the breeze and stuff like that, your root hair plexus picks up that. And then you do have the tactile disc, which you can see here, as opposed to just a free nerve endings, it's basically more like a disc end, right? So, and here's your root hair plexus. For the capsulated one, the encapsulated receptors, you have your end bulbs, um, you have your lamellated corpuscles, and you can see your basically connective tissues wrapped around them. This one detects pressure, this detects pressure as well, but we're talking about low frequency versus a high frequency. And you have your bulbous corpuscles, which detects pressure. We're thinking deep pressure as well, right? And it's a tonic receptor. You have your Meissner corpuscles, which discriminative light touch. Basically, this is how you can tell the difference between textures, shapes, and stuff like that. So you can see structurally the difference between them. This is your end bulb. You can see lamellated corpuscles. You can see the bulbous, and you can see the tactile corpuscles. Referred pain. I usually ask questions about this. I think I mentioned this in the last class we had, and it's basically a pain that's inaccurate, or the localization is inaccurate, I should say. So you're feeling the pain somewhere, but the source of the pain is somewhere else, right? So the brain basically didn't get the correct location of where the pain is coming from. So when you look, we talked about clinical relevance, but when you look in here, you can see different regions where you could have referred pain. If the pain is somewhere here, you could have the pain down your hand and stuff like that. Phantom pain. Phantom pain is basically a case where you have people who do have like pain in a limb that's been amputated, right? It's no longer there, but for some reason they still feel the pain. And this is usually based on the fact that the cell body is still alive, right? So the cell body, here's my fancy diagram, you know, all the whole dendrites and the axon, synaptic knob. But the cell body is basically this part is still alive. So you could have had like a cut somewhere, and this looks like a stick man. But this part is still alive, and it still thinks it's getting signals from other stuff, and that's why it could be painful sometimes. Well, painful even though the limb is no longer there. All right, we're just gonna go over different senses quickly. Like I said, I'm going over this slides. It's quicker, just with the focus points being on what I would typically test in an exam. So we have olfaction, which is the sense of smell, right? So you detect odorants, and an odorant is basically something dissolving in your ears, right? You have the molecules. I always joke about this. When if you can smell it, the molecule is in your nose. So you know what I mean. If you're in weird places and you're smelling weird stuff, you have that weird molecule in your nose. All right. So apparently it's kind of hard to do stand-up comedy when I don't have you guys standing in front of me. So the odorants, you typically have this, right? So stimulus, when we look at the ears, we look at the eyes, there's a form of stimulus coming in. The goes in, and then you have like the receptors, right? The receptor organ, and it typically distributed into three parts. You would have the receptor cells that detects the stimulus. In the case of the olfactory, detects the odors. You have the supporting cells which sustain the receptors, and you have your basal cells which basically the stem cells, and they replace the receptor cells every so often, right? So in the case of your olfaction, it's every 40 to 60 days you have your receptor cells being replaced. So I do like to ask a question about that. How how many days does it take for the basal cells to replace the factory receptors, something like that, which would be like 40 to 60 days. And all right, so other parts of it, which is basically you do have your lamina propria, the Bowman glands, and this is just like other parts that contribute to it. So the lamina propria, you have your areolar connective tissue, um, with one of the blood vessels, and you have your olfactory glands, which helps with like mucus to cover the epithelium, or like your nose, the receptor area. And as you can see here, the molecules go in there, gets picked up by the olfactory hairs going through your receptor. You have, and then it goes all the way to your olfactory bulb, which is then connected to your tract, which goes to the brain for interpretation, right? Talked about the hairs, the nerves, which of course helps with interpretation, and taking that information to the brain. And you have your bulbs, which is talked about, tracks. All right, nothing much here to talk about. So I'll talk about this briefly, and then I'll just like go over it with other stuff. Like basically, the point I want you to remember with this is the process where you do have the stimulus getting transduced, right? So information comes in, the other ones bind in, and they go in, and they lead to some form of depolarization, which is the key part, right? You're just going in and influx and outflux, sodium, potassium, in this case calcium. Something leads to depolarization, which then takes the information to the brain, right? The secondary neuron picks it up, and you guys remember the pathways, and that's where all this comes into play. And the same thing applies for like taste, sight. So I just need you guys to remember the process leads to a depolarization. Nothing major here. All right. So taste, like I said, you have gustatory, similar. Well, first of all, before we look at the taste buds, I need you to look at the tongue as a whole, right? So the tongue has different papillae. You basically have. I need you to remember which of them has or doesn't have a taste bud. So you have the filiform papillae, which is the only part that has no taste buds. You have your fungiform papillae, which is has a few taste buds, right? But the one that has the most taste buds is your valet papillae, which is considered the largest, has the most taste buds, and your foliate papillae. It's not well developed, but still has a few taste buds. So technically, the only one that doesn't have a taste bud would be your filiform papillae. So it has no role in gustation. And structurally, you can see the different structures, your filiform, your fungiform, and your foliate. And you can see here your valet papillae, right? So for this as well, you do have the gustatory cells, you have your supporting cells, you have your basal cells, right? So same concept, receptor cells, except like unlike the nose or the factory, this one you have 7 to 9 days before it gets replaced by the basal cells, right? And of course, you have the supporting cells as well. All right, nothing. No gustatory cells. We're just talking about the microvillas, just like you have the hair for the nose. You also have like the microvillas, which is like your taste hair. Next slide, please. Come on. There we go. All right, similar idea, like I said, with the pathway, takes the information, converts it, action potential goes to the secondary neuron, and then goes through the pathway to where it needs to be interpreted, which when we're talking about sensory, we're basically talking about the parietal lobe. All right, so you usually have sweet, salt, sour, bitter, umami, which are the five basic tastes, right? Umami being amino acids, the mini flavor. That's why everything tastes like chicken. Just kidding. Um, but what I really need for you to remember besides that is the tastings, right? So for sweet, bitter, and umami, the tastings are actually molecules, right? So the tastings bind to the cell membrane receptor, the molecules, and you have a G protein activation, second messenger, and then result in a cell depolarization, right? But for salt and sour, they're ions, so by themselves they depolarize the cells directly, right? So I would ask questions like, which of this is has like ions for the taste ends or molecules, so something like that. Pathways, similar we talked about. What? All right, these are fancy diagrams: the smell, the taste. So, of course, by now you should know most times when you taste food, you actually smell it as well. So, if you were to block your nose while you taste food, the taste will be totally different from what you used to, right? So, I think there was a study. Well, actually, I think it was Brain Games where they had a couple of people who were really good with like chefs or something like that. They blindfolded them, closed their nose, and have them taste the food, and they could not identify what food it was. All right, accessory structures of the eyes. By now, you guys should know the extrinsic eye muscles. You know, you have your eyebrows, eyelid, eyelashes. Based on the lab, basically, you guys should know the parts of the eyes already. Um, let's just go to the picture and talk about it. Not this one, this one right here. So you can see your eyebrows. You can see it, the eyes, the lens, which you guys should have seen. Should have your iris, um, the pupil, and of course your iris that helps change how much light that comes in here. You should have your conjunctiva, which once again helps so you don't have stuff like contacts going into your head like some people think it does. You have your aqueous humor in here. You have your vitreous humor on the inside here. The ciliary muscles, you should always remember they're right here. You do tend to see those on the lab test, so make sure you remember those. You have the lacrimal apparatus, which basically produces, collects, and drains the fluid, right? Which, let's see here. You can see lacrimal gland in here. You can see the I want to say dumping sites, but explains why when you people cry something, the nose starts to run, right? Because that's where your lacrimal sac is. All right, the eye structure. I need you to remember the three tonics, right? You guys probably seen this already in the review. You have the external, the middle, and the internal, right? So the outermost parts is the fibrous, which basically constitutes of your sclera and your cornea, and you do have your vascular middle parts. We're talking about your choroid, the retina, your neuronal, pigmented parts, and we're talking about like neuronal with rods and cones. We'll look at that here in a second. But I just need for you to remember all three parts, right? Which is the innermost one, which you want to say outermost. Ideally, I would say you should be pretty much familiar with this diagram, right? Even not for the lecture, for the purpose of the lab. Off the bat, I'm not sure, but I want to say you probably get a good amount, like about probably 10 questions just off the eye, maybe, yeah. Oh, a couple of good questions, yeah, probably up to that off the eye. So make sure you definitely familiarize yourself with it. Both this and the extrinsic eye muscles as well. So we talked about the fibrous, which is the sclera. So the white part of your eye is the sclera, and the cornea, of course, is the transparent part that covers your eye. So once again, like when you do like a laser surgery, this will be the part that the cornea that gets like taken out. Well, in the case of PRK, completely. In the case of like a LASIK, it just gets cut, flipped open, and whenever the surgery is done, you just flip it back. In the vascular tonic, you have your choroid, you have your ciliary body, and the iris, which basically is where your eye color comes from. You, I don't believe I have it on the test, but every once in a while I do like to ask the question, where does your eye color come from? And of course, controls the diameter of the pupil, which is the opening of your iris where light comes in. So you have your sphincter, pupil muscles which constrict, and you have your dilator pupillar muscle which basically dilates. Um, of course, you have photoreflex which just responds to light accordingly. So you can see here constricted versus dilated. So for the retina, you do have like the pigmented and neural layer, right? So this is where light basically gets refracted onto by your lens, and when information gets picked up. The pigmented layer is attached to your choroid, which has all this blood vessels and stuff, and it also helps absorb stray lights to prevent the light from scattering. And your neural layer is basically where your photoreceptors are housed, right? So you're talking about rods and cones, where one is for color and the other is not. So rods and cones. You have your photoreceptors, and then you have your bipolar cell layers, and then you have your ganglion cell layer, right? So almost like layers from one to the other. Takes the information, goes all through this, and then down there. I think I have a picture somewhere here. And before I show you the picture, the optic disc is basically where you have no photoreceptors. So this is what we typically consider your blind spot. So if someone is trying to get into your blind spots, they're basically trying to get to the point where whichever light is refracted off them falls on the optic disc where no information can be found. You have your macula latae, which is basically um, contains your central pit, the where you have most cones, right? So there's not much rod here to this part where most of the color is being interpreted or picked up. And you have your peripheral retina, which contains mostly rods. So we're talking about areas of low light. So this just gives you like a diagram. See the choroid, which we're talking about, and then you have your photoreceptors, and you have your bipolar cells, ganglionic cells, and it goes in like that. Same stuff. Well, clinic detached retina, in which case you basically it could come from a head trauma where the retina gets detached, and also could be caused by, I mean, led to by diabetics and someone who's already nearsighted. Let's go to lenses. So ideally, you guys should know by now the lenses change shapes. You probably saw it in the lab to be super hard, but that's because the cows, well, the eye is dead already, so it's solidified. But ideally, your lens is enough to change shapes to refract the light, depending on the distance away of what you're looking at. And we talked about the 20/20 vision, meaning at 20 feet, you can see something normal people see at 20 feet, and if you have like a 20/30 vision, it means it's worse than 20/20. So what normal people can see at 30 feet away, you have to be 20 feet away to see it. And if you have 20/10 vision, it's way better. And saying what normal people see at 10 feet away, you can see from 20 feet away. All right, so ideally you have this, you see the stuff, and then it goes refracted right there, which is why whatever you actually seeing, it's seen. Well, the information gets to your retina upside down. So whenever your brain interprets stuff, it has to interpret it upside down, right? So every time you see stuff, just know your brain is seeing stuff upside down. Um, and like in the case where you have like short-sightedness, myopia, hypermyopia, the image is either overshooting, ending up here, or not getting there, right? So that's why you have corrective lenses to help with that. So in the case where you're far-sighted, right, it means you can see stuff far away, but when it comes close, you can't see it, right? So let's say you're far-sighted, you can see this like normally, but by the time it comes closer, by the time this refraction ends, the object will end kind of here. Well, kind of. That's where it's projected to end. And the problem is it's not on your retina, so your brain can't get the information. So you would need like a corrective lens, contact, or glasses that would basically do, well, yeah. So you have the shape where it's going to for bring refract it a little more before it hits the lens, and by the time the lens does its own refraction, it should end up here. And in the case of nearsighted, you get the contact lens or glasses that does the opposite. All right, cataracts you basically have with aging, comes with aging, same as glaucoma. Well, typically both of them would come with aging, and this is where the lens starts losing clarity, reduce, and this could technically lead to blindness, but reversible, right? And the way to fix this is to take out the lens and replace it with an artificial lens. Talked about the vitreous humor and the aqueous humor, and both of them basically help with balance, right? So don't confuse the aqueous humor for tears, right? This is not where your tears come from. This is just that part that helps balance the pressure. You have the vitreous behind the lens, and you have the aqueous in front of the lens. Now, if you don't have like a proper balance, aqueous humor, we talked about this. That's what leads to glaucoma, right? Where the pressure builds up in your eyes, and that could actually lead to blindness. So you would have your aqueous humor here, and your vitreous humor will be behind the lens. I talked about glaucoma. Talked about visual impairment, where you have myopia, you have farsightedness, myopia, you have nearsighted, astigmatism, presbyopia, which is age-related. All right, refraction of light. And now you guys should know the difference between reflection and refraction. Refraction is basically a bend in the light, while reflection is basically reflecting it back, right? So, oh shit, that line was not straight, but the reflection just reflects back, but refraction would basically the light comes in and bends, right? So that's why you can see this looks different. Alright, not so much to focus on here. Talked about this. Alright. So for this transduction, once again, transduction, we're talking about converting a stimulus to electrical signal. So for the transduction, we'll be converting light to electrical signal, right? So let's look here. We're talking about rods and cones because they're basically the photoreceptors you have. I do like to ask questions on rods and cones. I'm not sure if I put multiple. Yeah, I did put that there. So the questions I like to ask is which one is for color, and that's cones, right? And rods is more like a black and white. Um, they can distinguish color. Another question I like to ask is which is more abundant, right? So you should know that you have more rods than you have cones. And we have they all contain opsin protein, right? Which is basically for the pigment. So you do have the based on the type of opsin you have, they do detect different wavelengths. So you can see here the blue cones would detect a short wavelength, but the green cones do the intermediate, red cones long wavelength. So you have something like this right here, right? And so depending on what's being detected, different parts or different opsin is getting activated, which is how you differentiate color. And color blindness is an excellent recessive condition. Basically, not everyone gets it. It's mostly found in males because, of course, you know you're male. Um, Males have the XY chromosome, and females XX, which means if you're colorblind, let's say you have a C here. Okay, that's not what I meant to do. But let's say since it's a recessive condition, right? If a male were to have a recessive, and I'm supposed to put a C here, right? Okay, let's do this. So let's say a male, like if you have that color blindness, a recessive, yes, recessive, right? So if your one X chromosome has the C because of the male only has one X chromosome, even though it's recessive, there's no other chromosome to like override it. So if a male were to have this chromosome, they're definitely gonna be colorblind. But in the case of a female, if they even if they have one of it and the other one is normal, it would override it because it's dominant over this, and that's why most males have it. But that doesn't exempt females from having it, right? Because if, let's say, someone the father is color blinded, and the mother, even though she's not, has a recessive chromosome that's colorblind, that encodes for color blindness, right? And if they have a daughter, but the X, of course, if you have a daughter, this X chromosome definitely comes from the dad, and this also comes from the mom. So you're going to end up having two X's, which encodes for a female, but both of them would have a color blind. So which makes it the female color blinded. So if someone who's a male who's color blinded marries a female that has a recessive gene for color blindness, definitely the whichever if they have a daughter, she has a 50% chance of being colorblind. But of course, if they have a son, it's gonna be the same because the father only gives the Y chromosome to make a son, and the mother gives one of this, so it's gonna be 50% chance as well of having a color blinded son. All right, enough of genetics. Let's go back to you. Uh, Anatomy and physiology. So cool part about the rods, it's in the dark. They basically assist retinol. If you've taken organic chemistry, you should know the difference between cis and trans. But they all the in bright light they're trans, which basically is a dissociated form of upstaining, which we call bleaching, right? So every time you go from light to dark, dark to light, there's a change in conformation, right? So almost think of it like you have to rebuild the rods when you go from light to dark. Right? So if you do have this right here, so you're in bright light. The moment you get into bright light, the conformation changes. You can see a cis to a trans retinol. So see change in the conformation here. Now, when you go back into the dark, it has to get restructured basically into the cis retinol. And the process it takes there is what makes your eye have to adapt, right? So you go into a dark room from a bright room, your pupils dilate to take in as much light, but your rhodopsin is trying to get the conformation to happen. So that whole process takes time, and before you get a full adaptation to the dark, it could take up to 20 to 30 minutes. But in the case of light adaptation, it takes probably about 5 to 10 minutes because you don't have to go through that same process. All right, optic nerve tracts. We're just talking about the same concept, picking up the information. Nothing much here to talk about. Let's see. All right, ear structure. For the ears, similar concept as well. We usually have the outer ear or the external ear, middle and inner ear, right? With the external ear, we're talking about the auricle, which is basically the part of your ear we can see, the helix and the lobe that makes up that. You have, of course, your tympanic membrane, what you call the eardrum, which vibrates based on the sound. We have the middle ear, the eustachian tube, and this is kind of contributes to pressure equalization, right? So usually when it's closed and you yawn, allows air movement and basically balances that out. So what else was I gonna say? No, nothing else. So the auditory ossicles, which are the ear structure, the middle ear as well. I do like to ask questions on those, right? Like which of this is not or which of this is. Make a habit of knowing both names, right? So the ideally we're called malleus, incus, and stapes, but based on what they resemble, sometimes they're called the hammer, the anvil, and the saddle, right? So for this, just make sure you know what they are. I typically ask based on these names, but like I said, remember the structures as well. And auditory ossicles, but once it goes through the middle ear, then you end up going to your inner ear. And I'm gonna go through this quick as well. What's the picture? So what I really need, you just remember that in your ear you have your cochlea, you have your vestibule, and you have your that region, and you have your semicircular canals, right? And this is where all the nice action takes place. Are you sure I'm gonna ask questions on this on the exam? But it's just good to have a general idea of the structure of your ears. Let's see. Just going through this. I think there's just one more part I usually test you guys on. Not this. Well, oh, yeah. So just look at the hair ear here, and remember this. I want to show you. I'll show you something about balance here in a second. But for the purpose of the exam, remember the difference between pitch and loudness, right? Question always says, what does it depend on? So the pitch depends on the frequency of the vibrating object, while loudness depends on the wavelength, the wave amplitude. Loud, the wave amplitude is what determines loudness. So I usually ask one or the other. I know I put the multiple choice on there, but just know the difference between both. All right, in the pathway, similar concept. You know, basically trying to get a depolarization. You can see deafness. You could have conductive or sensorineural. One is basically an interference, and this can be easily fixed. But this is usually a permanent type of deafness. All right, so equilibrium. I was going to show you this macular no once. Uh, no, yes. So when you look in here, you can see this is basically part the way the brain also tells the orientation of your head, right? Remember, your ear contributes largely to balance as well. So based on the directions of your stereocilia, which is the hair, if you want to put it that way, tells your it tells the brain basically which direction your head is tilted, right? So this would be normal where the head is straight, but by the time you bend your head, you can see how that orientation changes here, right? So changes from that to this, and now your brain is saying, "Oh, now I know your head is tilted back." And when it's this way, my pressure puts it here, and I'm saying, "Oh, your head is tilted forward," right? And when you think about like dizzy spells, this is what where it gets interesting, right? Because when you have a dizzy spell, your brain is basically confused. So like someone goes on a boat, so people who like feel dizzy on a boat, your eyes is seeing something and your brain is seeing something else exactly. So it's moving in one direction because your brain is saying, okay, your eyes are seeing this, but the ear is responding to this, and then your proprioception is out of balance, basically, and that's where dizziness comes in. All right, I think, well, for the purpose of the exam, yeah, I think that was it. Yeah, that's all I really have for you guys for the exam. If you have any question, please email me before the exam, hopefully. Um, and I hope you guys do well on the exam. All right. Thanks for your time. Have a wonderful day.